Today we will discuss about doxycycline derived gene editing, control over gene editing. So in this video we will discuss how we can use this doxycycline derived some plasmid that can control the gene editing that is going on within our cells with the help of Cas9. So this is one of the examples uh, that use doxycycline as a activating as a activating promoter for this Cas9 G7 guide RNA. So in normal case, when the doxycycline is not activated, this Cas9 G7 is not translated, as we can see here that it is inhibited in the normal case, and because of this the Cas9 that is present within the cells will be is able to continue its gene editing. However, when the doxycycline is introduced into the system, what it will does is it will remove this inhibition of this TET-O, and the translation of this TET-O to TET-R will translate this Cas9 G7 guide RNA, and this Cas9 G7 guide RNA will then cleaves off this Cas9 at the G7 site, and thereby this editing of the further editing of the within the cells will not occur whenever this doxycycline is introduced into the cells that are transduced with this plasmid. So this approach provides us a more customization control over the experiment. So in this case we can make a uniform in this case we can produce a more biological replicates where those results are close to each other, because we have a better control over the duration of time for which this gene editing will be will be carried out within the cells, as there is no variability that can be observed at the time of if we use some other techniques such as electroporation to transduce this SG guide RNA. So in that case, there is the one of the variability could be the efficiency of the electroporation within the different kind of libraries or cell lines, and other variability that arises is the as different cell lines have their own time to recover from the electroporation. And so these are some of the variabilities and that arises when we use the approach that does not have control over this gene editing time period. So the one of the best advantage of this approach is to provide this gene editing control, so that we can uh, precisely control and the time period for which the gene editing will be carried out within the cells. So this doxycycline will, when it reduced, usually this Cas9 contains the different region of this guide RNA. They are named as guide RNA 6, 7, 8, 9, 10, and this paper use the guide RNA or this G7 Cas9 G7 guide RNA, so that it can cleaves off the G7 side of the Cas9, not the other side. So this is what happens when this translation occurs. When this translation produced, then it will cleave off. It will go to this Cas9 site, that Cas9 genome present within the cells, and it cleaves off. After that, this cells has their inherent capability to repair the DNA, and one of the approach by which they repair the DNA is this NHEJ, that is non-homologous end join. So cells will try to repair this DNA breakage that occurs because of this guide RNA that cleaves of the Cas9, and this DNA repair, there is a more probability that DNA repair will not re-establish the function of Cas9, as this DNA repair is completely random based on the analogous pairs of the two gene edited site. So this is just an example, or one of the example by which we can have a more better control over the experiment, or over the, more specifically, over the gene editing time. If you need to understand more about this approach and some of the results that are present, if you want to see some of the results that are present using this approach, you can go through the reference that has been provided in the description. And one more thing that arises if we have a long gene editing time period is that this Cas9 will also perform some non-specific gene editing because of the increased duration of the gene editing time. So it is essential to have a precise control over gene editing time. So that's all for this video. Thank you for your time.